really good to be here in what I consider my second home, Texas. I'm from Eastern Canada, right on the Quebec border. It's good to see some friends of mine from Last Days Ministries, Joan and Roland here. And actually, my start in creation science, believe it or not, started right here in Texas, in Lindale, Texas, at Last Days Ministries. And I was over there working with Roland and Joan and about 250, 300 people, something like that, other people there. And we had this guy by the name of Carl Baugh came by, and we had a television studio, full television studio. And this guy came by and did a 5.5-hour lecture off the top of his head. Now, I had, after backsliding from the Lord for quite a number of years, I returned to the Lord. And when I did, I was wound up down at Last Days Ministries, and I had taken on an interest in creation science, but I had never heard this stuff before. It just floored me. I couldn't sleep for days. And it really prompted an interest. So this man right here is really, aside from Jesus Christ himself, the creator, this man is the reason that I'm here today and involved in creation ministries. And I do want to say thank you for everything you've done and all the support. So I'm glad Dr. Bob brought up these because, I want to start off with something a little simpler first. I want to settle a little discussion here. Some might argue that there is no such thing as a creator God. I would like to present proof to you that there is a creator God. And I will start off with this. Do you think that chair had a creator? You seem so sure of yourself. Eh? It was built. Really? Oh. She seems really convinced. Is anybody else convinced that chair had a creator? A number of you are. All right. I'm going to come back to that, okay? Now, Dr. Bob brought up the gills in fish. This ties in heavily with 5 models I brought along with me. These are simplified models. There's a reason I simplified them. These are models of various hearts. Now, you got to remember, according to evolution, we started off as lowly protobacteria, evolving into fish, to lungfish, amphibians, reptiles, land mammals, birds, etc. Now, there's a really big problem with that. Now, this is a simplified model of a fish heart. The reason I made these simplified is because the real thing is so complicated, it is almost impossible to model, and even more impossible to understand when you just take a look at a model. So I've simplified these dramatically. A fish has a simple 2-chambered heart. The blood returns from the body, deoxygenated, into here, goes through a one-way valve into the pumping section, which then pumps it out to the bodies. Down in Texas, we say out. It's spelled O-W-T. We spell it differently in Canada, so I hope you'll be patient with me. It pumps it straight to the gills. Now, there's a very important point to be made here. You notice, I'm sure most people here are aware, that humans have 2-chambered hearts, a low-pressure side and a high-pressure side. A fish can get away with pumping high-pressure blood straight to the gills because it lives underwater. You see, in order to get maximum gas transfer, you want those blood vessels to be as thin-walled as possible, but in order to make them as thin-walled as possible, it makes them weak. With a fish, because it lives underwater, the water pressure provides a back pressure on those fine blood vessels, so it can pump straight high-pressure blood right to the gills, maximizes its gas transfer, and then sends that oxygenated blood to the body, then returns to the heart. Are you with me so far? Okay, good. Okay, now that's a fish heart. The frog heart is just a little bit more complicated. Again, a simplified model. The frog hearts have 3 chambers. They have 2 returns from the body and the skin, which come into these 2 chambers here. Both those chambers share a common valve, which goes into the pumping section. The pumping section actually takes that blood and pumps it out to these 5 blood vessels, but you see that little squiggly valve right there. That is really important because what's happening here, you have 2 levels of blood. One is very deoxygenated, one has a little bit of oxygen left in it. as it comes around, it is what engineers call a laminar flow. They don't mix. What that little valve does is it keeps the laminar flow intact, and it diverts the deoxygenated blood to the lungs and skin, and the blood that has slight oxygen goes straight to the body. It then cycles back and comes back to the heart again. It's a 3-chambered heart. Now the crocodile heart, this one is really interesting. Probably one of the most complex hearts out there. It is a 4-chambered heart, much like yours. It's got a low pressure side and a high pressure side. Okay, so blood returns from either the lungs or the body into the atriums. That goes into a one-way valve into either side of the heart, which then pumps it through the vessels either to the lungs or to the body. The low pressure side goes to the lungs and is low pressure so it doesn't burst the capillaries. Now, crocodiles have a couple of very unique features about them. Number one, and you get a chance later on when you get the chance, come up and take a look at these. Okay, you get a closer look. This valve right here is unique to crocodiles. It actually has a cogged tooth. So these two cogged gears actually mesh together, lock together, and they do not allow blood to flow out through that to the lungs. Why does it do this? Anybody know? I'm just curious. No? You don't know?
Yeah, I guess.
Okay.
So that it can submerge in the water for extended periods?
You've got it. You've got the nail on the head. That's exactly it. What it does, it actually shuts off blood flow to the lungs. Why? Because when it's underwater, the lungs are useless. The lungs will actually rob oxygen from the blood. It actually shuts off the lungs, and that allows it to stay underwater for up to 2 hours at a time. Anybody want to try and compete with that? No? No takers, eh? Okay. I used to snorkel a lot. I used to be able to hold my breath for 2 minutes underwater. That's a long time underwater, let me tell you. 2 hours. I've heard some reports of up to 8 hours. I don't know if I believe it or not, but that's what they report. Now, the other part is right here, what they call the foramen of Panizza. That is a vessel that joins the two sides of the heart. That works in conjunction with that valve. Because what it does is it bypasses the lungs now completely, allows it to stay underwater for long periods of time. Very complex system. So now let's move to the human heart. It's similar to the crocodile heart. You have 4 chambers, again, because we want as thin-walled capillary in our lungs as possible, but if we pump high-pressure blood to the lungs, it would destroy our lungs. We would bleed to death in our lungs is what would happen. It would kill us. So again, our hearts are 4-chambered, a low-pressure side and a high-pressure side. Now, there's something that very interesting that happens during fetal development in the womb. And this comes from our good friend, Professor Amy Clark, up in Illinois. He was the one that put me onto this. Now, Professor Clark pointed out that babies in the womb, they don't use their lungs. They receive the oxygen from the mother. So God has created and designed a system in which it allows the heart to pump with both sides of the heart to the entire system of the body. The reason it does that is because the right side of the heart, when the child is born, that heart needs to be fully developed and ready to go. If it isn't, the baby's got a lot of problems. It's not going to survive. The Creator has designed something absolutely remarkable into this. There is a vessel which you had when you were in your mother's womb, just like the crocodile. It's across the two sides of the heart. It also has an opening between the 2 returning chambers. What that does is that bypasses the lungs, and the right side of the heart, which is the low pressure side, pumps along with the left side to help in the development of the baby. The other amazing thing here is the blood returning from the body is completely deoxygenated. The blood returning from some of the vital organs has some oxygen. whereas the blood returning from the umbilical cord has high oxygen. The creator is an absolutely remarkable engineer. Absolutely remarkable. Professor Clark is a hydraulic engineer or a hydrologist, and he's quite convinced the creator was a hydrologist as well. I'm a robotics engineer. I think he was a robotics engineer, but hey, I think mathematicians think he was a mathematician. Anyway, the creator has designed this valve which stays open during development, and right here is what they call the crista dividends. And as you can see, it diverts oxygenated blood into one side of the heart and deoxygenated blood, which then goes to the umbilical cord. Again, a laminar flow. Now, sadly, in this fallen world, what happens once in a while is during birth, what happens is the left side of the heart becomes the high pressure side. What happens then is it actually closes that valve. That valve, for all intents and purposes, actually heals closed at birth. This vessel right here is made of a very special muscular type of material, which when it's exposed to high amounts of oxygen, actually shrivels up and closes off. All of this happens at birth. If it does not, you wind up, sadly, with what we call a baby with a hole in its heart. And a lot of times it kills the baby. I've had friends who have had that still open as an adult. They get into running and whatnot, and it's very difficult for them to run and breathe and provide oxygen for their body because of that. Now, this is an excellent example of why it is absolutely impossible for any of these hearts to have evolved. You got to remember, this is supposed to have evolved through small processes and small steps into this. It absolutely cannot happen. What happens if you poke a hole in one of these major arteries to develop lungs that don't exist yet, such as in the lungfish? You kill the organism. End of discussion right there. You kill it. The cardiovascular system of the bird, which is very similar to our hearts, except the lungs in the bird. Now, I worked for an engineering company up in Pembroke, Ontario. We made what we call cool suits. And you may have actually seen them. I sure wish I had one today. But what we made there is basically a suit that you wear with these veins of tubing running through them. We run cold water through that. If you actually see the Space Shuttle astronauts suit up, just before they put on their final outer suit, you will see them, they have on our suit. That's our suit, believe it or not, manufactured in Canada. Eastern Canada, used in NASA. Well, when I was working for that company, what we do is we engineer the heat transfer very carefully. The creator has engineered the bird lungs very carefully. In order to allow maximum gas transfer, the blood and the lungs actually run in opposite directions. Very carefully engineered. The lungs are not like ours, they're not bellows. The air comes in one end, goes out the other. Anybody here knew that already? Nobody knew that already. Really? Oh, come on. I thought they taught that in high school. Okay. Okay. What's that? Yeah, okay. Air comes in one end of the lungs, goes out the other. The blood, low oxygen blood, comes in one end, goes the opposite direction. We do the same thing with hot water transfer and cold water transfer. It maximizes the oxygen transfer. Our creator is an amazing engineer. Absolutely amazing. You think that chair had a designer? Yeah? Okay. Well, I'm going to move down a little. What I want to talk about is the DNA. This is a computer program. Essentially, it is a program that it is in, we believe, every single one of your cells that is instructions, it's a code on how to build you. You are a unique individual. Nobody else has your identical computer code. They have a lot of similarities, and all of life is based on this computer code. Now, there are 4 what they call bases. 4 bases, Adenine, thymine, guanine, and cytosine. These are 4 chemical compounds which join together with a sugar backbone into the helix, which makes our genetic code. That's found in all your chromosomes, for instance. You're with me so far? Okay, good. I just wanted to make sure everyone's awake here. This Texas heat, boy, I tell you. Last night, Stan and I, we enjoyed our motel room. They had real good air conditioning. Oh yeah, we had it down to around 64 degrees. It was just right. We like it cold. Now, this computer code, which is very careful instructions on how to build you, is very specific. If there is one error, as little as one error, if it's a deletion of one of those lines of code and it happens to be in the wrong place, it deletes the entire gene of that part of the DNA. Genetic errors are deadly. Absolutely deadly. Now, how many people have heard it claimed that chimps and humans share 98.4% identical DNA? How many people have heard that? A number of people, yes. Now, believe it or not, that number, they came up with that number when they'd only decoded roughly 10% of the human genome. That tells you right off the bat, that should send up red flags to say, hold on a second here, guys. Something doesn't line up here. How they claim they achieved that, they lay out the genetic codes. Where the bases match, it is considered part of that 98.4%. Where they're different, it's considered the difference. Now, what does that mean? That 1.6% difference adds up to roughly, and you can do this on a calculator, the human genetic code is roughly 3.2 billion base pairs long. Think about that for a second. 3.2 billion lines of code. If you take that DNA and unravel it, depending on who you ask, you can get from here to the moon and back anywhere from 1 to 5 million times. Take the whole thing, it'll fit in a teaspoon. That's pretty good computer programming. That's impressive. But what does this add up to? Well, 1.6% of 3.2 billion adds up to roughly a little over 40 million changes in that genetic code that needs to transpire in order to turn a human into a chimpanzee. Now, as I've already stated, one change is enough to kill the organism if you're not careful. A maximum of 3 is all it takes to completely kill the organism. In humans, it causes nasty things like cystic fibrosis. But the change doesn't end there. Were you aware that the chimp genome is roughly 10% longer than the human genome? How many people knew that? Some of you would. Stan does. Yeah, Stan knows everything. He's from Washington. What can you say? It's 10% longer. Their chromosomes are tipped with a unique DNA sequence called the B7 complex. Chimps are unique. Humans are unique. But this brings up an interesting point. We can take a look at the genetic code, and incidentally, you can download all these numbers off the internet. A lot of the genetic code links are now available for free on the internet. You can do your own research from home. And I came up with a new theory of evolution. I based it on the length of the genetic code because more genetic code means more information. That makes sense, doesn't it? Okay, so I based my hypothesis on that. Algae starts off with roughly 541,000 base pairs in the genetic code. It's a very complex life form, but it was one of our first primitive life forms. Through random mutations, natural selection, accumulated mutations, we get up to the E. coli bacteria. I'm going to talk about that in a minute. At a base pair length of 4,639,000, it was only slightly more complex than the algae. And then you can see the similarities between the two, right? Then through other evolutionary processes, we wound up at the oyster, which has a genetic code 700,000 base pairs long, or 700 million, sorry. Way up here, we finally achieved the dog. 3.35 million base pairs, or billion base pairs long. From the dog, we evolved into the human, from the human to the chimp, from the chimp to the anteater. From the anteater, we finally achieved a miraculous step in the evolutionary process. We got as complex as the toad. From the toad, we got up to the red pine. Red pine is a vastly superior life form to you. Did you realize that? Based on the length of the genetic code, the evolutionary process does not line up. The amoeba would be classified as one of the most complex life forms ever in existence, with a genetic code length of 670 billion base pairs long. The amoeba is supposed to be one of the simplest life forms. It's all topsy-turvy. None of it lines up. Now, I mentioned E. coli bacteria. That is one of a number of bacteria that have this special propulsion system. Now, has anybody ever heard of a bacterial flagellum? Oh yeah, quite a few people. Bacteria such as streptococci and others have this tail which they spin. Depending on the bacteria, it'll spin anywhere from 20,000 to 100,000 RPM. Any engineers? We got a couple engineers. Anybody know what the speed of some of our modern jet engines get to? Pretty fast, eh? Depending on the jet engine, I know the F-15, which is without question one of the more modern jet engines out there, the F-15 jet fighter, that engine will only top out about 45 to 50,000 RPM. This is double the speed. Now, you got to remember, this is a biological entity. This is like you being able to take your arm and spin it around and around and around and around. How does it do this? It spins it in such a way that this hook holds it on an angle that it takes on a corkscrew shape, enabling it to propel itself forward in the liquids that it lives in. You follow me so far? Okay, this is very complicated stuff, I know. But let's take a look at this bacteria. Now I can't get it unplugged. There we go, engineering marvel, there we go. Let's take a look at this bacteria. What we're going to do is we're going to slice it up the middle and take a look in this area here and see what we can find. Here is the outer layer of skin. There's the outer layer of skin. This blue part would be the hook. So you have your outer layer of skin, inner layer of skin, and it has, get this, all the parts of an electric motor built into a little bacteria. We have the stator, the C ring, the M&S ring make up the rotor. We have the drive shaft, which goes up through the bushing or bearing, the L and P ring. It goes into what many have called the universal joint, the hook. It then drives that long tail known as the filament. Now, let's see if I can get this one working. It is quite remarkable how it does all this. The rotor is driven by protons, not electrons. And you've got to remember, this spins it up to 100,000 RPM. It's about 80% efficient. That's more efficient than most of our electric motors. Incredibly efficient with its energy. But recently in nature, there was discussion of this hook. That hook is actually controlled by the bacteria. The bacteria can change the angle of that hook on the fly. So in other words, it can actually change the angle of that hook while it's spinning at 100,000 RPM. This is the same as you being able to take your arm, put it around a corner, and rotate it at 100,000 times a minute. And actually able to hold that angle as it turns it. This filament, there was a tremendous amount of research done in Japan. That filament was actually made up of a series of protons. Now, or not protons, proteins, sorry. The proteins are joined together in a circle, 20 to 30,000 proteins long. How it assembles that is quite unique. It actually sends up, because you got to remember, these bacteria divide. So one of the lucky bacteria gets to keep the propulsion system. Okay, the other one, it has to make its own. Tough luck, you're on your own. Good luck with that, let me know how it goes. So here's how they do that. It's a hollow tube and this is sort of the outside cross-section, it sends up the protein through the center. This cap turned out to be very, very important. That cap has a series of legs which it can flex and is attracted biochemically. It leaves an opening exactly where that protein is supposed to go. It goes up through the middle. It is guided by that cap. It is guided into place where it is then polymerized or glued. When it's glued into place, that leg then bends over, is attracted biochemically to that one, leaving a hole for the next one. It does this 20 to 30,000 times to make up that filament. Now, you think that chair had a designer? You think that chair had a creator, huh? Do you think that model had a creator? You think so, eh? Okay. If the model that's based upon the real thing had a creator, why is it we are suddenly expected to believe that the bacteria did not have a creator? You see, the evidence of a creator is all around us. That chair had a creator. This is your intelligence recognizing intelligence. Now, I'm the founder and president of the Mensa International Creation Science Special Interest Group. Do you want me to repeat that? Okay. I won't repeat it. I won't say it 3 times fast either. I'm sorry. Basically, we are a group of members who believe that the evidence for evolution does not stand and that the evidence supports supernatural creation. That is the premise of the group. We want to stand as an example that there are intelligent people out there who do believe this. How many people have heard of the SETI project? S-E-T-I. Anybody know what SETI stands for?
Search for extraterrestrial intelligence.
You got it. Search for extraterrestrial intelligence. Now, here's an oxymoron for you. They are looking for evidence of intelligence in outer space. The search for extraterrestrial intelligence. Yet, when you bring up examples of complexity and design in life, they say, no, that didn't require intelligence. But then they go off looking for intelligence in radio signals. It is a contradiction to live the evolutionary lifestyle. It is an absolute living contradiction. All of life speaks of incredible complexity. Now, Stan, I want you to help me out with this. I'm going to close off with this. This is not a living cell. This is one protein. Now, it works out quite simply. There are 20 different amino acids to choose from. Your protein's 200 amino acids long. Therefore, it is 20 to the power of 200. Now, why don't you tell them, why did you come up with this, Stan?
Well, I've spoken on creation science for about 3 years, and I had just gotten done reading Francis Crick's book. Francis Crick discovered molecular DNA with his colleagues back in 1962 or 63, and I had read his book. And on Easter Sunday of this year, I was driving home from my parents, and I was praying. It was a long drive home, and the Lord said, well, why don't you write all these numbers down and look at them? So I wrote down 10 to the 260th power. This is the odds of an amino acid sequence happening by chance. Francis Crick also says
I just stand on this side.
that the estimated number of atoms or fundamental particles in the universe is 10 to the 80th power.
Somebody has a lot of time on their hands to figure that out.
So I wrote that down. And so the Lord told me to multiply out how many seconds of time there are in 20 billion years. So I sat down and multiplied that all out. It's a pretty easy thing to do. It comes out to 631 quadrillion, 152 trillion seconds in 20 billion years. And I thought, wow, if it takes that long, or if it takes this many odds or chances to get one protein, and there's only that many seconds of time in 20 billion years, divide the 20 billion year seconds into this. This is how many how many changes there has to be every second of time in 20 billion years to get one protein.
How many zeros is that?
Actually, it's basically 10 to 242nd power.
247. Okay.
242 zeros after that.
Okay.
And that's how many changes has to be every second. Why don't we see a change happening every second now? We don't see any changes. We don't see any changes happening that are not forced or man-made.
Excellent. Okay, thank you. I wanted his help on that because I still am not familiar with all the numbers. As you can see, they're very large numbers. But you can come up here and you can count the zeros if you want. 10 to the 246th? Okay. This is 42nd. Okay. This is how many changes are required per second. And Dr. Baum, maybe you can correct me on this, but to the best of my knowledge, we have not observed changes like that at all, if I'm correct there. And I mean, somebody obviously has way too much time on their hands when they have to figure out how many atoms are in the universe. But this gives you an idea of just how many changes and how complex life really is. The Creator is an incredible engineer. Just incredible.